So this is going to be about the lytic and lysogenic cycles in viruses, and these are basically the two main methods of viral genome reproduction in the host cell. So I'm going to start off with a quick introduction of viruses. They're extremely small, non-living particles between 20 to 500 nanometers. They're made up of only protein, including the capsid that protects the genetic material, and one type of nucleic acid. So it either has DNA or RNA, but not both. And then notably, they infect all living organisms, and they do not have cytoplasm, plasma membrane, or any organelles. Next, we'll be talking about host cell dependency. This is another key feature of viruses. They don't have organelles and cell structures necessary to replicate themselves, and they cannot reproduce outside the host cell. Thus, they rely on it for all of their functions, and so they are deemed obligate intracellular parasites. For example, they are dependent on the host cell for energy for viral replication. They don't have their own source of energy, so they rely on what the host cell provides through respiration. Also, nutrients, they do not have the ability to obtain their own nutrients, so they rely on the host cell for the necessary building blocks that they need for replication. Replication machinery, so viruses do not have the machinery they need to replicate their own genetic material. They use ribosomes and enzymes from the host cell to instead translate all their genetic material into viral proteins. And finally, transport. Some of the viruses actually use the host cell's transport machinery in order to spread to different body parts or other hosts and thus infect us. Accessing the host cell is another significant feature. There are two main ways that this happens, either receptor-mediated fusion or an endocytotic pathway. So in receptor-mediated fusion, viruses basically bind to receptors on the host cell, as shown in the image, and then they fuse directly with it and then inject the genetic material into the cell. This is different to the endocytotic pathway, as in that one, the whole virus cell is invaginated into the host cell. So the membrane invaginates in this process, and then the virus is able to fuse with it and enter the cell. Some viruses can actually be taken up by a host cell endosome. Now we will finally be talking about the lytic cycle. So this is the simpler of the two processes. What happens in this process is the bacteria or virus will attach to the host cell and insert its genetic material inside. The genetic material only enters the cell, and it basically forms a circle. Now, as stated before, it then uses the host cell's replication machinery to replicate this DNA or RNA multiple times until there is a large number of viruses assembled inside the host cell. Over time, this number of viruses will simply become too much for the cell membrane to hold. Remember, animal cells do not have a cell wall, thus they actually will lyse or burst after a certain point in time, and then the new viral particles are released from the cell. Now all of these viral particles can attach to new host cells and continue this process within the body. It's also important to note that in this process, the host cell is destroyed, which isn't the case in the lysogenic cycle. Now moving on to the lysogenic cycle, it starts with the viral DNA being injected into the host cell in pretty much the same way as in the lytic cycle. So both are the same to the point of DNA being injected into the cell. However, from then it is different. No viral particles are actually produced, and in this cycle, the host cell is not destroyed. In fact, it's generally a more complex and longer cycle because, as we see in the image, the DNA from the virus integrates with that of the host cell. This new circular DNA is now called the prophage. And as the host cell reproduces, each new cell will now have this bacteriophage genome called the prophage. Now, occasionally, as shown in the diagram, the prophase genome, the viral genome, does get excised from the host cell, and this forms bacteriophages and begins the lytic cycle. In fact, the lytic cycle can be triggered at any point in the lysogenic cycle and is often the case in many viruses, which can switch between the two different cycles when exposed to certain environmental conditions that can be either like UV light or chemical stressors. For example, the bacteriophage lambda, it uses both to infect the E. coli bacterium.